Hello again, this is Tanner and Maddie and Brayden. Okay, number one. We have a list of things. It asks transcriptional regulation bacteria involves a combination of regulatory proteins, DNA sequences, and small effector molecules. And then it gives us a list. So A is asking which of the terms describes or is an example of regulatory proteins. So for that, we found the CAP and then also activators and repressors to be regulatory proteins. For B, it's asking which of the terms describes or is an example of a small effector molecule. And for that, we got inhibitors, inducers, allolactase, and the CAMP. For C, it's asking which of the terms describes or is an example of a sequence found within the DNA. And for that, we have your +1, your -10 site, and then you have your LAC-A, LAC-Z, and also the operator. Then for D, which of the above is not involved in the LAC operon, and that would be the inhibitor. And then for E, which of the above is an example of a structural gene, that's both the LAC-A and LAC-Z are examples of that. And then moving on to 2, I'll also do that. Okay, so fill in the blank. For A, the LAC operon is regulated by 2 inducers, and that's allolactase and CAMP. For B, the binding site for sigma factor and RNA polymerase is called LAC-P for the first answer, while the CAP site is the binding site for the activator that regulates the LAC operon. For C, the enzyme that converts ATP into CAMP is adenyl cyclase. And then for D, the -35, -10, and +1 sequences are found within the LAC-P portion of the LAC operon. Then for E, the LAC-I gene encodes the LAC repressor, while the LAC-Y gene encodes lactose permease. For F, it says blank can regulate gene expression in bacteria by causing transcription to terminate prematurely, and that is attenuation. Then finally for E, an E. coli strain that contains 2 copies of the LAC operon is called a merozygote strain. Okay, 3, 0.
I got it. Not to speak different language.
I just did something.
Got it. Hey, there it is.
Did you stop it?
No, it's still going.
Still going. All right. So number 3. Let me look at my answers real quick. Okay, so for A, if IPTG is absent, glucose is absent, XGAL is present. Okay, so that means if IPTG is absent, the LAC repressor is bound to the LAC-O. If glucose is absent, sorry, they're all laughing at me, so I can't focus. CAMP levels are high, CAMP binds to the cap, and then the cap binds to the cap site, which means since the LAC repressor is bound to LAC-O, the LAC operon is not transcribed, and since that happens, the B-galactose site is not produced, which then means that the XGAL will not be cleaved to the blue, which means the E. coli will not be blue. So for B, if IPTG is present, glucose is absent, and XGAL is present. So if IPTG is present, the LAC repressor will be released from LAC-O, and then if glucose is absent, CAMP levels are high, CAMP binds to the cap, then the cap is bound to the cap site, just like the last one. When the LAC repressor is released from LAC-O, the cap is bound to the cap site so that the LAC operon is transcribed, which means the B-galactosidase will cleave to XGAL, which will cleave to the blue product, which makes the E. coli blue. So C, you got to hold on, there's 3 more of these. if IPTG is present, the lac repressor will be released from the LACO. So if the glucose is absent, CAMP levels are high, CAMP levels are high, then it's gonna bind to the cap is gonna bind to the cap site, and then lac operon is transcribed again. The B-galactosidase will be produced, but since there is no X-gal, it can't cleave to the blue, which means that the E. coli cells will not be blue. So D, IPTG is present, glucose is present, X-gal is present. So if IPTG is present, the lac repressor will be released from the LACO. Glucose is present, or the glucose is present, CAMP levels will be low, so it can't bind to the cap, and the cap will be unable to bind to the cap site. And when that happens, it is unable to bind to the cap site. The lac operon is not transcribed, and since the B-galactosidase is not produced, X-gal will not cleave to the blue, so the E. coli cell will not be blue. And then the last one, man, if IPTG is present, the lac repressor will be released from the LACO. If glucose is present, the CAMP levels will be low, and if they're low, it will not bind to the cap. The cap will be unable to bind to the cap site. And when the cap is unable to bind to the cap site, the lac operon is not transcribed, and since neither B-galactoside or X-gal is present, there's no blue for it to cleave to, so the E. coli will not be blue. And that is all for that one.
Okay, so for 4, so it gives you multiple different strands and their functionality. So the first one, all aspects of the strand are functional, so it is inducible. B, since it's IS, that means that the LAC-I, the repressor, is able to bind to the operator, but it's not able to bind to allolactose. So there's no way for the repressor to be taken off of the operator. So it'll be non-inducible. C, it'll be constitutive because the promoter in the chromosome is non-functional, but the promoter in the plasmid is functional, so expression will still occur. And then for D, I said non-inducible because the lac repressor of the plasmid is unable to bind to allolactose, which means that it will bind to the operator of the plasmid and the chromosome, and it will not be able to be removed. So just because that it's non-functional in the plasmid doesn't mean that it doesn't, since it produces the repressor that can't be unbound, it binds as well into the chromosome. And then for E, it's constitutive because the LAC-I in the chromosome is producing the defective or the repressor that can't be removed, but also the operon system, the operator and the plasmid is non-functioning, so expression doesn't change. They kind of cancel each other out. And then for 5, it gives you, again, various different functionalities of the different aspects of the lac operon system. And it gives you lactose or lactose positive environment and a lactose negative environment, and asks you to determine if there would be a color created. So a 0 is no color, 1 is that one copy of the lac operon is transcribed in 2, so an amount of yellow, and then 2 is again yellow coloring, but when both copies of the lac operon are transcribed. So for the first 2, there's only one strand or one copy, so there could only be 0 or 1. So for A, the promoter region is non-functioning, so in the lactose positive environment, it won't function because it's not able to be started because the promoter is not functioning. And then the lactose negative, obviously, since there's no lactose, there will be no function. With B, the operator is non-functioning, it's defective. So in the lactose environment, it will show expression because it's not able to be turned on or off, I mean. And in the lactose negative, it'll show expression, because even though there is no lactose, there is still, it says glucose levels are low, but there's still some glucose, which means there is still some CAMP, some CAMP that can bind to the cap site. And then for C, the lactose positive, it'll have both strands are showing expression because the first strand is fully functional and the second strand, it's just the operator that's non-functioning. And then again, in the lactose negative, one strand will be functional because the operator is non-functioning, so it will not be turned off. And then for D, in a lactose environment, one strand will work because the lac ion, the first strand, is defective, and so it won't produce, it won't have any expression. And then the lactose negative, since the operators are both functional, then they won't show expression. And then for E, the lac I's on both of the strands or copies of the lac operon system are both unfunctional, and the lac S, that's what produces the repressor that binds to the operon or operator and isn't able to bind to allolactose, so it won't show expression. And then, so it's 0 for both of those, and that's it.
That's all, folks.